everybody for being here. It's so exciting to see all of your faces that I haven't seen in such a long time. So that is so nice and heartwarming. I wanted to start out by telling a little story about one of my first days in a grad school class here. And I don't see him here, so it's okay, I won't offend him. So I was in John Drake's population ecology class, which was terrifying. And he asked a question. His first question of the first class was, what is ecology? And this is an easy question. I'm going to get my hand out there and I'm going to answer this one first. So I said something, and he was like, wrong. So over the years, I'm not sure if I've learned what ecology is, but I really like John Muir's quote here that I think summarizes for me what ecology is, and it is the integration of all of nature. And I like this. It says one tugs at a single thing in nature, he finds it attached to the rest of the world. And I think that is what ecology is. It is learning about how all of our systems are integrated, humans, wildlife, nature, how it all comes together and forms a whole. Further problems?
No.
Yeah. Maybe click. Mine's too slow. Okay. So the title of my dissertation is very broad, and that is intentional. It covers, honed in on these three topics: ecological, genomic, and social factors that impact wildlife conservation. And so this covers the human dimensions and some of the natural sciences. It ties into my previous work in molecular biology with the genomics, and then pulls in some ecology, looking at environmental impacts. And so I'm in ecology and part of the ICON program, and I just wanted to take this opportunity to thank John, my advisor, the committee, Lizzie King, Nate Nibelink, and Bram Tucker. But I also wanted to point out and call out special thanks to Vanessa Zenwa and to John Gittleman for a lot of input over the years into this entire project. So endangered species are an interesting topic because in order to tackle the conservation problems that surround endangered species, we have to look at a lot of different facets of what goes into that conservation. One is engaging with humans, and that's just to put very simple terms. But this is essential for positive conservation outcomes. And this is one of the primary reasons that I joined the ICON program, is because I knew that I had training in the natural sciences, and I knew that I could do molecular biology and contribute to conservation, but I did not know how to engage with humans in order to promote positive conservation outcomes. Another is that endangered species have lower genetic variation than non-threatened species, and so we know this. There have been surveys done, and so on average they suffer from lower genetic variation, which has implications for fitness and for disease, which I'll talk about later. Lastly, they're more likely to live in degraded environments. And so this also has impacts on their fitness, both direct and indirect. So throughout my dissertation research, I've tried to tie these themes together throughout my chapters. And so one of the chapters that I undertook was to look at the social context of conservation. And I did this by doing interviews in Samburu, Kenya, which is in the northern region of Kenya. And I looked at three factors that are often misaligned between Western conservation ideals and when we do work in developing countries. And those are knowledge perceptions, locus of control, and the motivations that underlie conservation. And I'll go into more detail when I talk about that particular part of my research. Another thing I'm particularly interested in is disease. And we know that genomic variation in wildlife has a huge impact on disease. But we see variation in these association across all studies. So we see lots of difference in the outcomes across studies. And so one way to get at answering this question is to pull together all of the empirical studies and do a meta-analysis to see what trends are across systems. And the last part of my study is looking at the environmental impacts on fitness. And for this, I did field research in central and northern Kenya. And I did this on the Grevy's zebra. And so looking at the environmental correlates of both stress and infection in this species. So my the first part of the study, I'd like to delve into the social context and the human dimensions a little bit. So wildlife conservation is not solely an ecological problem. It really entails negotiating this interface between people and wildlife. And we hear a lot about this, I think, in terms of human-wildlife conflict, in terms of these direct interactions that people have with wildlife. But there is a lot more that goes into it. It's also understanding people's motivations, it's understanding the agency that they feel in undertaking conservation, and it's understanding how they perceive the environment that they live in and the wildlife species that live there. And when we're talking about international conservation, whether that is aligned with what people in developing countries, how they perceive their environment. The title of this particular study was Knowledge Perceptions, Locus of Control, and Motivations Regarding Wildlife and Wildlife Conservation in Samburu, Kenya. Sorry, I'm getting some internet requests. So international conservation is often based on Western-derived ideals. And when I say Western-derived ideals, I'm talking about Europe, US. And so three of those things that I honed in on are that from these Western ideals, we often look at rarity of a species and the possibility of extinction as primary reasons that we are going to conserve a species. And so these are really things that are promoted. The species is rare, we need to take care of it. The species has high likelihood of extinction. This is based on, oftentimes, on the red list, the IUCN red list, which categorizes as endangered, and critically endangered, and vulnerable, and those are extinction categories, or extinction risk categories. Another thing that is a primary component of Western ideals for conservation is that humans are both the cause and the solution to wildlife conservation. So while we can be detrimental to wildlife populations, we also hold solutions to helping wildlife conservations persist and increase in number. And lastly, this is prevalent. So in developing countries, conservation NGOs will come in, often international conservation NGOs, and promote conservation with economic incentives. So this is either through direct payment, it's through tourism, through supplementing families with school bursaries, but in some form of economic incentive is the way it's promoted. And so I touched on this before, but when these Western-derived ideals are used in developing countries in conservation messaging, they may not align with what the people who live there locally and who are experiencing the impacts of living with this wildlife are perceiving. So I did the interviews to ask whether people's beliefs about wildlife and wildlife conservation align with these Western ideals in central and, well, in northern Kenya, in Samburu district in northern Kenya, which is, if you're familiar, so this is Kenya. This is actually outlining the Grevy's zebra range because it's the IUCN map for them. But this is Lake Turkana, and so it's just south of Lake Turkana. So the interviews involved asking communities if they identify with Western-derived ideals of conservation. And I did 74 one-on-one interviews with both men and women, tried to find equal numbers of men and women in these areas, and basically touched on those three key areas that I touched on before. So the first one was knowledge perceptions. And for this, just to key in on this a little bit more, we asked about abundance of wildlife trends and how Samburu are perceiving wildlife trends over their lifetime. And then we asked them, how are you perceiving wildlife trends currently, in this current period? And I'll go into a little bit more detail about what I mean by those time frames. And then finally we asked them if extinction is a possibility for a species. Secondly, asked about the perception of agency in terms of wildlife conservation. do they feel that they have an impact directly on the ability of a species to persist there? And so this is locus of control, and so we asked them about this in terms of the impact that they think that humans have, that individual themselves have on wildlife, or whether they don't, or whether it's under the control of something like fate or God, or something that's out of their hands. And the last one was, we asked, what are the primary motivating factors for wildlife conservation in Samburu? And we asked people to free list that, which is we just asked them about what motivates them to conserve wildlife, and had them list whatever they wanted to, and then we grouped it into economic and non-economic incentives. And finally, for the last one, we asked if species rarity is a motivating factor to provide more care to a species. And so I just want to go into a little bit of detail because this will become important. So the interviews were conducted in 3 community conservancies in Samburu. So Mebai, Kalama, and Westgate. And so community conservancies, just briefly, are areas that have been sort of in combination with governmental and private partnership, sort of cordoned off, if you will, for conservation. So they're pastoralist areas, but they've been sort of they have a fenceless border, and inside of that fenceless border, they have certain restrictions on what the pastoralist can and cannot do in terms of grazing their livestock, or in terms of densities of livestock that they can keep. And there are often incentives promoted to be a part of these community conservation areas. And so importantly, these 3 areas differ quite a bit. And what I wanted to highlight is that the difference in the tourism assets and in the presence of conservation organizations. And when I say conservation organizations, I mean primarily ones that are promoting the Western ideals that I discussed. And so I don't expect you to remember these, but I'll go back through it. But just in terms, so the major differences would be between Westgate and Mebai. So Westgate has a lot of tourism. They have, if you've heard of Save the Elephants, they have a Save the Elephants camp there. They have a lot of other organizations operating, and they have many tourist lodges. So they see a lot of tourists coming. They may perceive benefits from having tourists come, in economic benefits. And Mebai, on the other hand, is more remote. It's not closer to a national reserve where they're getting a lot of wildlife coming over, and they do not, as of now, have any tourism assets, and there are very few conservation organizations that are actively working there. Okay, so all of the interviews were conducted with Samburu. And Samburu are primarily pastoralists. And I know I mentioned pastoralism before, but just to briefly update or to talk about what that is. It's a lifestyle that depends primarily on raising livestock. And so given that the area, as you can see, where Samburu pastoralists live, they move quite a bit in order to find grazing for their livestock. And so that sort of informs their perception of the environment, this movement with livestock. They are moving regionally to find grass and water for their livestock. And we conducted interviews with men and women, and I just wanted to spend a little bit of time because this will become important. So Samburu traditionally don't record the exact date of their birth. Instead, you're born into an age set. And so you can think of age set something like millennials, baby boomers, something like that. And so typically you join an age set, men join an age set when they're circumcised. And so this is a ceremony that I would say, on average, happens around the age of 13. So these years refer to when an individual is about 13. It's a little bit more difficult to gauge how old women might be because they typically take on the age set of either their husband or men who are of a similar age. Okay. So the first one for perceptions of species abundance and extinction. So I talked about this, but local conservation partners, they might form perceptions on different spatial and temporal scales. And again, we travel widely, but this is not true of most of the Samburu pastoralists. They have mostly stayed within their small area. So they might not perceive these ideas of rarity or extinction and be informed or be, have that knowledge sort of information stream coming to them about rarity is important and extinction is important in the same way that we do. But importantly, sharing these knowledge perceptions can lead to improved conservation outcomes. So there have been studies that show that actually taking information from people who have more fine-scale information on species rarity and joining that with traditional natural science information can improve the outcome for conservation programs. So the first thing we did is to take a table where we had the age sets, and again, this is just an approximate age, and we had 10 stones, and we asked people to put the 10 stones on the table representing what they thought the trends in livestock abundance were over their lifetime. And so, for instance, I just want to highlight this. If you were a Likashili, you would only answer for the age sets that you had experienced. Similar, if you were a Lukaroro, you would only answer for the age sets that you had experienced. And then we recorded this as that they had increased, decreased, or fluctuated. And then for the current age set, so this is beginning in about 2006. We didn't interview anybody from this age set because they're typically, well, they would be under the age of 18. But we did ask people what was happening in this current age set. So increasing, decreasing, or staying about the same. So most Samburu interviews had perceived that wildlife abundance had been decreasing over their lifetime. And this was significantly, there was a significant skew towards the belief that abundance had been decreasing. Whereas during the current time period, a significant shift in that most people thought that wildlife abundance is increasing. And notably, there were differences here between the conservancies. And again, Westgate was the one that had the most tourism and the most input from conservation NGOs. And this is the place where, this is the community conservancy where most people believe that wildlife were increasing. So when we asked about extinction, most people believe that extinction is not possible. In fact, when we ask the questions, extinction is not an idea that is familiar to most people, and so we would just explain it as there would be no more of this species left in the world. And that generally did not resonate and was a concept that most people couldn't seem to conceive of being possible. And so again, Westgate stood out here, and that more residents of Westgate expressed that extinction was possible. And just again, this is the one that has the most input from Western conservation organizations. When we asked about locus of control, which is an individual's perception that his or her actions or his or her ability to bring about change through their behavior. So the way we did this is when we asked about the abundance trends through the lifetime, we then asked people to say, what do you think was the cause of these abundance trends? So whether they said it's increasing or decreasing, we just asked them, what do you think caused this? And they free listed the reasons, and then we grouped those into human and non-human related causes. And so human related causes would be something like poaching, killing for bushmeat, and non-human related causes or disease are fate or something out of human hands. And so remember, for the lifetime abundance, most people thought that wildlife was decreasing. And so, but this is attributed primarily to humans and a combination of human and non-human causes. And then during the current time period, when most people believe that wildlife abundance is increasing, this was also attributed to human causes. And so here you see this sort of alignment with this traditional Western-based ideal that humans are responsible for both the increases and decreases, or both trends in wildlife population. And again, Westgate stood out as being the place where more people attributed changes to humans than to non-human causes. And so finally, when we asked about motivations, and just reminding everyone that economic incentives are the, sort of the main sort of paradigm that Western conservation organizations promote for conservation in developing countries. So we asked people to free list what they rated as the reasons to conserve wildlife. And I wanted to highlight that, in fact, so a lot of studies have looked at the using economics as a motivating factor for wildlife conservation, and shown that when that is used alone, when economics and incentives are used alone, it actually has a negative impact. And there was this lion study done in Amboseli, Kenya, that looked at either economic incentives alone or the combination of cultural and economic incentives, and found that when culture and community values were incorporated, there was a higher success rate to disincentivize lion killings than when only economic incentives were used. And so for the motivations, it was mostly a combination of economic and non-economic reasons, or economic reasons alone. And only 2 people cited non-economic reasons, which were aesthetic reasons or things like they've always been here. And so there were only, and I found this very notable, that only 2 individuals elicited these responses, that there were, the reasons were purely non-economic. And again, Westgate stood out here as being the community conservancy where economic reasons were more prevalent than in the other areas. And finally, I'm not going to show a graph for this, but we also asked explicitly about rarity and whether people cared about whether a species was rare. And the way we asked about that was to say, would you value it more? Like, would you provide more care for it if it were rare? And this, I think, is a familiar topic to a lot of people there because they see conservation organizations coming in and providing individual care to animals. And so we're asking, if you know this species is rare, does it deserve more care? And less than 25% of the people said that that reason alone, rarity, is a reason to provide more care to it. The rest of the individuals said it should receive the same amount of care as every other type of species, or they said, no, it shouldn't get any care. So I wanted to wrap this part of the study, by saying, just sort of summarizing the results. And the way, one of the take-home, sort of takeaway messages that I took from this is looking at these differences between community conservancies and the possibility that people's perceptions of how they value wildlife, so their knowledge perceptions, whether they think rarity is important or extinction is possible, whether they think wildlife should be kept around for money reasons, for economic reasons, and whether they have agency in wildlife conservation, might be being influenced by these Western conservation organizations. And so what we saw is that wildlife, the prevalent themes were that wildlife is increasing and is due mostly to human-related reasons. And this was in, these were the overall answers. And that extinction, and from Westgate, sorry, and that extinction of a species is possible. And that economics is the primary reason to conserve wildlife. And so just tying this back to the factors that are associated with each of these conservancies, I think it's an unanswered question that we know that that's what's causing people's beliefs to differ between these community conservancies, but I think it's a potentially very interesting avenue of research, especially given what we know about changing people's perceptions and valuing wildlife. And that, in fact, promoting economics incentives may not be good for long-term conservation success. So although we're seeing short-term alignment with Western ideals, it may not be good for the long-term outcome. So the second part of my study. So I'm very interested in genomic variability. I think there are so many unanswered questions, especially in wildlife non-model systems, about how genetic diversity contributes to fitness, how it contributes to population viability. There are just so many unknowns, and we talked about some new unknowns yesterday. Endangered species are interesting here because they are more likely to have lower genetic diversity than non-threatened species. And so this has implications for fitness, it has implications for disease. So this chapter, I did a meta-analysis, and I was interested in understanding the factors that contribute to differences that we see in this association between genomic variation and disease across wildlife systems, and I just focused in on mammals for this. So typically the prediction is, right, as we see increasing levels of genomic variation, and I will get into more detail about what I mean by genomic variation in a bit, we see increasing levels of fitness. So we see increasing survival, increasing reproduction, which is the definition of fitness, as genomic variation is increasing. And I guess maybe I should restate that. Individuals with higher levels of genomic variation have higher fitness. And that is the general prediction. So it's very difficult to measure fitness in wild populations that have long generation times. So I'm working in the Grevy zebra for my field system. It would be very difficult to measure survival and reproduction over a PhD term. And so one way to look at fitness is to use disease as a proxy. And so we do know that a lot of wild populations have suffered from disease. In fact, it's contributed to population die-offs. So the saiga antelope is one example where we had mass die-offs of this species. And then a lot of people also know about chytrid, which has caused big die-offs in amphibian populations. We also know, this link between genomic variability and disease, and that just as genomic variability impacts fitness, low genomic variability can have a negative impact for disease. So the typical prediction here would be that individuals with lower levels of genomic variation have higher levels of disease. And this is because both resistance and tolerance decrease as you have decreasing levels of genomic variation. And this has implications at both the individual and the population level. So individuals have greater levels of disease, they may have worse outcomes once they get infected, higher levels of infection. And also, it has implications at the population level because we see increased transmission. So the Tasmanian devil is one example where there's very low genetic variability at a major immune region. So the major histocompatibility complex, the MHC, and I'll refer to that quite a bit throughout. And so this has led to an 80% die-off in the Tasmanian devil population because they are unable to block this transmissible cancer from infecting them. So we see and we predict that low levels of genomic variation can lead to increased disease, worse disease outcomes. But across systems, we see these sort of equivocal results, right? In some cases, there is, when there is higher levels of genomic variation, we see increased disease. In some cases, there is no observed relationship. This doesn't mean that there isn't something going on there, it just means we're not measuring it. So just a few empirical examples. So not all animals with low genomic variation show signs of disease. So in the northern elephant seal, they have very low genetic variation at the MHC, which is, again, this immune complex that I talked about, which is one of the most studied areas involved in disease, but no detected disease. And in feral cattle, again, between individuals, near uniformity at MHC, but no detected disease. And then on the flip side of that, there are some animals where we know have very high genetic variation at MHC regions, but also have a high incidence of disease. So what is going on here, and what can we predict based on measuring genomic variability in wildlife systems? What can we predict about whether they're going to be impacted by disease or not impacted by disease? And so to try to get at some of these factors that are influencing this association between genomic variability and disease, I did a meta-analysis, and I collected empirical studies in wildlife, in mammals. The main steps here were to conduct a literature search. So I used 3 broad databases to search the literature, and then extract study data from all of these publications. And then by extracting the various ways that people had presented their data, to convert that into a standardized effect size, and also extract the effect direction. So the literature search, I used all terms that I could come up with, and this was based on scanning a few other abstracts and things like that I knew should be involved once I did my literature search. All things that would include sort of genomic diversity, genetic diversity, heterozygosity, et cetera, and came up with about 6,000 articles. So from those, I just did a title, scan through, and got rid of things that I knew didn't fit what I was looking for, which were things that did experimental infections or involved domestic animals or other studies that were not wildlife and not natural infections. And so that actually honed it down to 36 studies in total, and each one of those had several lines of data, either because they studied multiple different parasites or they did it in different seasons. And so they were in the final data set, 295 lines of data. So I wanted to spend a little bit of time talking about the direction of effects here, because I think it can get a little bit confusing, and it has gotten confusing in conversations. So across studies, what we did when we pulled out the data is we recorded the effect direction, and that was, if you have higher genetic variability, do you have a better outcome for disease? And that would be actually a negative association, going back to our graphs. I'll just pull this up. So if there's a negative effect size, higher genomic variability equals fewer parasites. If there's a positive effect direction, higher genomic variability equals more parasites. And I'm calling them parasites here as a sort of catch-all term. And so we pulled out the effect direction, which was generally just stated or given in the statistics. And then, depending on how they reported the association between genomic variability, we calculated that into the standardized effect size, which is the magnitude of the effect. So the first thing that we asked, is there a mean effect across studies or by phylogeny? So looking at the mean effect across studies, we just took all of the studies and we said, is there a general direction that all of these go? Is there a general size of the effect? And the answer to this, the predicted answer would be no, because we already know this, the reason we're doing this is because we see this variability in outcome between the association of genomic variation and disease. So there was in fact 88% heterogeneity in effect sizes across all of the studies. So there was no common direction or common effect size of genomic variation on disease in wild mammals. And then, somewhat surprising and somewhat disappointing to me because I am interested in this avenue, is that there was also no effect of phylogeny or of species relationships on the association between genomic variability and disease. And there were 28 unique host species that were included in the study across all 36 of the papers that we pulled out. So given that there's no mean effect and that it's not affected by phylogeny, what are the factors that explain this variation across studies? And to do this, we used a moderator analysis. So this is just looking at the factors that vary between studies. And we just honed in on a few. Primarily, how people measure genomic variation varies drastically across studies. And so this is kind of the primary question here is, how does this impact the association? And then how is disease measured? And then what parasite taxa were measured? So genomic variation can be measured in multiple ways. One, and traditionally genome-wide heterozygosity has been used, or genome-wide diversity. So this is done at putatively neutral markers, and this is typically done as a proxy for inbreeding. And people have seen associations between this and increased parasites, so higher inbreeding, more parasites. You can also look at heterozygosity, which is the difference between the alleles at a given locus at loci that we know are particularly related to disease. So that's just looking at one or a few loci. And the last way that we pulled out is people can look at the presence of specific alleles, like the identity of this allele. Is it present or is it not present, and what is the impact on disease? And then for that, it was in our study, it was all MHC. So the first one, for the first test of moderator variables, we grouped heterozygosity, just looking at genome-wide heterozygosity and heterozygosity at specific alleles. So we just grouped those together and we said, okay, heterozygosity as a whole. And then we looked at specific alleles, and we looked at the differences between those two, and saw that they have very different impacts. So there is a significant difference between the association between genomic diversity and disease, between whether you use heterozygosity or specific alleles in the study. And so while specific alleles have an overall positive effect size, meaning that there are more, it was when there was a presence of specific alleles, there were more parasites. And then, as expected, when heterozygosity was higher, there were fewer parasites. But that is not very meaningful on its own. Okay. And so we chose to hone in just on heterozygosity and say, well, we know that there are these two very different levels of heterozygosity. So one is just looking across the genome and just looking at inbreeding effects, and the other is looking at these regions that we know are linked to disease. And so the expectation would be that this would have a bigger effect because we know that these have an impact on disease. And so for this, we looked at the candidate gene heterozygosity, which are the disease-related genes, and we looked at neutral, which is just genome-wide diversity. And then there's another way to measure diversity, which is the actual base differences between two different alleles at a specific location. And for this, there was no significant difference of effect between the three ways to measure heterozygosity. But candidate gene heterozygosity overall did have a negative effect, or fewer parasites when there was higher levels of candidate gene heterozygosity, which is not surprising. Fits with expectations. And then we wanted to look at specific MHC alleles on their own, because alleles can vary in their frequency in the population. And oftentimes rare alleles will be more associated with a better disease outcome because parasites have not evolved. And so the animal with these rare alleles is still able to recognize and combat the parasites. So we looked at the frequency of alleles in the population and broadly categorized them as being rare or common. And this was based on reported frequency in the paper. And I did test different cutoffs for rare and allele between 8 and 20, and there really is no variation between there. It's significant independent of cutoff of the frequency. And so rare alleles have a negative effect size, meaning that, unsurprisingly, as predicted, that when there are rare alleles present in an individual, they have fewer parasites. So this again fits with predictions. So we're not finding anything that's very surprising in these studies in terms of genomic variation. So candidate gene heterozygosity and rare alleles are correlated with lower incidence of disease. Finally, the last thing we looked at, and we'll go over this quickly, is that we looked at different ways that studies measure disease. So we looked at infection status, are you infected, yes, no, parasite burden, parasite richness, which is the number of parasites in an individual, and then disease-related illness or mortality. And none of those were related to the association between genomic variability and disease, nor was parasite taxa. And that was independent of how we classify parasites. So we did it as micro, macro, extracellular, intracellular, which I thought was an interesting thing because they elicit very different immune responses. So I think the fact that we found no association says something about the genomic variation that we're measuring, and not that there's probably nothing there. So to wrap this up, so I think this highlights, so our findings weren't surprising, that we get more information with candidate genes, and we can see something when we look at the differences in alleles. But inbreeding and the loss of adaptive variation are both important for conservation in wildlife. And so one thing I think that this study highlights is that, we might be overlooking inbreeding effects because we're not, up until now, we haven't been able to capture enough loci to really understand inbreeding effects and to really look at genome heterozygosity. And so given the increases in technology, I think maybe we will be able to include more putatively neutral loci and capture this. And the other thing is that all of the studies involved MHC. And so in the study that I referenced earlier, where there were high levels of MHC diversity, but there was no disease measured, this could very well be due to the fact that we are just not looking at variation in other regions of the genome that are involved in disease. So I think, again, with increasing technology, this is something that will be very possible. I'm going to speed through the field chapter. So the last study that I looked at, and maybe my favorite, because it was a phenomenal experience in so many ways, was the work that I did looking at the environmental effects on stress and infection in Grebbe's zebra. So so the environment, so similar, I'm going to show some graphs similar to what I did with genomic variation. So we know that environmental degradation has as it increases, fitness of species can be negatively impacted. So I looked at this, the effect of environmental correlates of stress and infection in the Grebbe's zebra. So Grebbe's are endangered. They are a species of zebra that is only found in central and northern Kenya and small pockets in Ethiopia. I just looked at the Kenyan population. Their population has dropped from about 14,000 in the 70s to about 2,500 now. This is from about 2007, so they're actually might be increasing a bit, but it's stabilizing somewhere around 2,500. And in addition to this, precipitous population drop. They also have undergone extreme range contraction. So this in the light green is their previous range, and this is their current range. So they used to cover, in almost the entire Horn of Africa, and now they're just in this small region of Kenya and even smaller part of Ethiopia. And so one reason that it's really a good system to look at berry zebra in Kenya is because it is this matrix of land use types, and that is due to climate reasons, and it's also due to historical reasons in the way land was divided under colonialism. And so there are different regions, and so these are some of the environmental factors that we measured. So there are areas that have, these are goats, have very high densities of livestock. There are regions that have very low densities of livestock, and that is generally where you see more wildlife, generally. There are also differences in, the way people are using the land. So some regions have really high densities of pastoralist activity, and then other regions it's mostly tourism and/or research activities. In conjunction with this, there are differences in the amount of water that we see from area to area. I grouped these because these tend to go together. So it tends to be, areas where there's high livestock, high pastoralist, there's little water. Areas where there's more livestock, more tourism, more water. And the other factor is forage that goes along with this. So I did field work in central and northern Kenya in 2014 for about 4 or 5 months, and then in 2018 for about 8 or 9 months. And this is just a picture from some of our field work, lab tent, counting parasites on a bench. And sampled Grebbe zebra across their core range. So this gray represents their core range. This represents the areas where I sampled. And this was generally restricted here because there aren't very many Grebbes here, and this area is a bit too dangerous. And so we collected fecal samples, and for each of the fecal samples did stress hormone levels, and then did fecal egg counts, so counted 2 types of parasites, gastrointestinal parasites. And I will show you this really quickly. So I just wanted to give you a taste of what the field work. So we would look for Grebbes. You can see some. You see these white butts. And if we could not get to them, close to them, we would get out, kind of track them, hang out, wait until they would provide a sample for us. Oh, they're running away. And so I just think that showing the video gives a better feel for what it was like. It was great fun. And so we collected the fecal samples and measured stress and infections. Stress hormones were measured as fecal glucocorticoid metabolites. I'm going to refer to them as FGCMs. And stress hormones in fecal samples provide a better indicator of chronic stress than like blood samples, which are more temporal, or a more immediate stress impact. And then we counted to those 2 types of parasites that I mentioned. All right. And John had a specific question about how much fecal samples. So this is just, so we would find the fecal samples, surprisingly difficult sometimes, given the amount of, the area looks the same. So we just collect it as much as possible without getting dirt and other things. So then we would mark the GPS location where we collected this, which was as close as possible to where the actual animal was. We collected these like within 30 minutes of seeing the animal, defecate. So they were all fresh. And so we know that previous studies have shown that Grebbe zebra habitat choice is impacted by environmental factors. We know that they choose areas away from livestock. They drink only at night when livestock aren't present, even though this has increased risk for them. But we don't know, how all of these factors are influencing their physiological fitness. So the reason that stress and infection are good measures to use are because we know that they are directly linked to fitness. We also know that they're impacted by the environment. So as an example, so wildebeest in East Africa had higher stress hormone levels when there was human disturbance and less vegetation. And same thing with cattle. When you have when there were degraded rangelands, they had higher intestinal parasite burdens. So we know these have been linked to environmental factors. And we also know that both increased stress and increased disease have lead to decreased reproductive success, decreased survival, and in the case of stress hormones, decreased immunity. So I'm actually going to, so the environmental correlates that we pulled out were water, because this can be a very limiting resource, anthropogenic activity, and for this, we separated between pastoralist activity and tourist activity because they are so distinctly located on the landscape. And then use normalized difference vegetation index, NDVI, as a proxy for the amount of forage. And these pictures that I'm showing just kind of give you an idea of the very different nature of some of the landscapes. So for these correlates, what we did is we drew a radius around where each sample was taken and extracted them either from ArcGIS or from the NDVI was from MODIS. So in terms of the buildings and the pastoralist homesteads, I mapped those in ArcGIS and we pulled out the densities of those. And so those were combined into a principal component analysis, which pretty much separated along the lines of high livestock, low road density, high road density, high tourism. So we had a pretty good separation between pastoralist activity and tourist activity. So the two questions were, how does environmental context influence stress hormones? And how does it impact gastrointestinal parasites? So for stress hormones, we saw increases on both ends of the range. So this is high tourist activity. This is high pastoralist activity. So although there is a lot of variability in the sampling points, you see higher levels of stress hormones with both types of anthropogenic activity. In terms of water, when there was no water, there were lower levels of stress hormones compared to when water was present. And this is a donkey, it's not a... and then how does it impact infection? So even though the strongyl nematodes had 100% prevalence, meaning that they were in every single individual that we sampled, we didn't detect any difference based on environmental context between the burden or the number of eggs that were in an individual. Lungworms only had a 57% prevalence, so we looked at this as infection status. Are you infected? Are you not infected? And did find that environmental factors impacted infection status of lungworms. So what impacted the lungworm infection status were access to water. So when there was no water, individuals were more likely to be infected, and to a lesser extent, NDVI. So when, and this was a correlate of the amount of forage or food available. So when there was less food available, they were also more likely to be infected. So I think the interesting takeaways from this are, one, traditionally in these conservation studies of wildlife, we've looked at pastoralist activity as having a very detrimental impact on wildlife. And this, I think, shows, and we've looked at tourism as being beneficial to wildlife conservation. So this, I think, shows that maybe we need to start to incorporate, right, some of the tourist activity and perhaps equate that more so with also having an impact just, as the pastoralist activity might. Secondly, I thought it was interesting because even though grevies are an arid adapted species, we're seeing this response to lack of water that may not be expected. And I think this has implications, right, for climate change scenarios because we're going to be seeing less water availability. And so even there are no other studies in wildlife, to my knowledge, that look at the direct impacts of water limitations on stress, and so I think this is potentially an area that would be interesting to look at as we see more drought and dry lands. I'm actually going to skip over this. And I'll just go to my acknowledgement slide. So again, I want to thank my committee and everyone that has helped me throughout. Tons of people helped in Kenya, so much. And then everyone that has helped me here and provided support and friendship and, ears and all of that. And all the funding sources that I had, which I could not have done, this work without that. So I know that we are very tight on time, but if there are any questions, I'd be happy to take them.
So, if pastoralists have a negative effect on where zebra, on zebra, is that mostly through the people or through their animals and the relationship there?
So that's a good question. I think that we don't know the answer. So what I measured in this study was livestock density and the density of their homesteads. And so the actual density of people was a little bit more difficult to get those actual numbers. So I think that would be an interesting avenue. I think it's probably complex. I don't think it's related to, in the case of zebras, I don't think it's related to their livestock impacting the amount of forage available. I think it would, because the other studies have looked at competition in that way, I think it would more be related to an actual sort of disturbance effect in terms of direct stress sort of impact. But I think that's an unknown. Yeah. Yep.
From the interviews that you were doing in the 3 different communities, do you have any information on Western style tracking of the populations of different wildlife types in those areas to see whether or not the local people were more aware of what, that they had a better on the ground sense of what was really going on?
Yeah, no, I think that's super interesting. And I think I don't have, so there are general trends, right, for all of those populations, but I would need to have sort of fine scale tracking, and so I don't have that information to do that, region and see if they correspond to what natural science or Western conservation picked up. But I think that would be very cool. Yeah.
I was wondering if you had thoughts on a controversial topic in Kruger is watering holes, where they promote tourism, and also maybe alleviate some of this water stress.
Right.
So is there watering holes in this area of Kenya?
Yeah.
Should we not?
Yeah, there are. So there are dams that are created. There are also some watering holes at the tourist locations where they, try to promote this wildlife viewing. I didn't put it up here, but one of the other differences that we did look at is the difference between water source types and their impact on stress, and actually found that near the major river in the area, there were higher levels of stress hormones in grevy zebra. Compared to other areas where there was water, like dams. And so, my take on that is that maybe that's a competition with other wildlife sort of effect. And so I think, maybe there are effects of drawing these, larger groups of wildlife in that might impact stress physiology that we're not taking into account. Yep.
So I guess back to your first chapter, so the locals at Westgate, how integrated are they with tourists and especially the conservation groups? Is there a lot of kind of interaction there that could influence perception more?
So in terms of tourism, so I'll do them separately. So in terms of the conservation organizations that are there, so that's primarily the headquarters of the Conservancy, and they are very integrated. So they're very active. They're going out in the community oftentimes. There are also conservation organizations like the Grevy Zebra Trust, who I worked with, is there. They are going out all the time, and they're doing seminars, and they're doing workshops, and so there is just generally a lot of integration there. in terms of tourism and that integration, a lot of people are employed in the tourist industry there, and so in that way, there's also a lot. So I would say there's more integration between those different spheres, if you will, in Westgate than I saw in other conservancies. Yep.
Probably just have one more question.
Yeah. Yeah.
Yeah, I was wondering, so when you're talking about the lungworms and the
Yeah.
spread like parasites,
Yeah.
so I know like in like livestock, you know how like they say like worms are found in like the bottom inch or so of the grass, and then if like
Yes.
you always rotational graze, they don't spread.
Yeah.
And you had said something about the lungworms and correlation with the lack of water. Do you think it has more something to do with like the ponds and stuff not being there, or the fact that maybe there's not rain there to make the grass grow this equal to there not being ponds there? You know, like, do you know or?
Yeah. So I don't know is the answer. But I would say that. So I didn't go into this, but I would, potential sort of reasons that we saw that impact are because, the stress of traveling further to water, I would say, right, that has an impact on an animal's energy, and therefore the amount of energy you can put into immunity. So when you're near an area with no water and you have to travel far, you're expending a lot of energy, so you might have decreased immunity. So it might have to do with like energetic trade-offs, energetic balance. But in terms of the environmental impact of the presence of the parasites in the environment, I couldn't take a guess on that. Did I get your question right?
Yes, ma'am.
Yeah, okay.
All right. Well, thank you, Sarah.